Welcome to BioSumit friends. Today we are going to learn about fluorescence microscopy. So as the name suggests, fluorescence and microscopy. So here actually fluorescence is used to visualize some specimen. Okay, suppose this is an object. This can act as a light source or it will be stained with some fluorophores. Fluorophores means fluorescent dyes. Okay, or it will be tagged with some proteins which can emit light when excited by some specific wavelengths. Okay, so when the object is being shown in a magnifying order in microscopy, so we can call it as fluorescence microscopy. So what happens for fluorophore and when you can say that fluorescence is happening? So fluorescence phenomena is, suppose this is an object, it will absorb a specific wavelength of light, suppose X, and it will emit in a different wavelength of light, that is Y, and Y will be greater than X. In that scenario we can say that fluorescence is happening. So for any object, when you are watching this in microscopy, so it can be we all that setup is called as fluorescence microscopy. Okay, there are some commonly used fluorescent dyes are there, like fluorescein, okay, or rhodamine D, okay, or DAPI. Okay, fluorescein, what happens, it actually emits some green light, okay, when excited with blue light. And rhodamine actually emits red wavelength when will be excited with some green light. Okay, so this is the phenomenon of fluorescence. Now for fluorescence microscope, it has actually three basic components. One will be excitation filter. Let me try to draw. Suppose this is excitation filter. One will be excitation filter. There will be one dichroic mirror. This is dichroic mirror. And another thing will be emission or barrier filter. Okay, obviously the object is here. Suppose, see, the dichroic mirror is actually situated with an angle of 45 degree with the object. Okay, here the angle is higher, I have drawn, but remember that is 45 degree. Okay, so here will be the lens obviously, and here will be the eyepiece. Now this excitation filter, suppose excitation filter is actually inserted in the light path between the light source and the sample. Okay, how it is worked? Like excitation filter will actually acts as a band pass filter. Okay, suppose there are some type of lights, I mean green, blue, specific wavelengths actually. So excitation filter is actually allow some specific wavelength of light. Suppose it's allowing right now is green. Okay, this is the excitation light, you can call it excitation light. Okay, so it will be reflected by dichroic mirror and it will go to the object. Okay, so the object is right now is absorbing the wavelength of the green light. Any light it can be, I'm just supposing it green light, so it will emit some longer wavelength, suppose red. Okay, so it will emit red wavelength. Now as it is longer wavelength, it can't be reflected by dichroic mirror, it can't be blocked by dichroic mirror. Okay, so it will pass through the dichroic mirror. Okay, now there is one emission or barrier filter is there. Okay, earlier the dichroic mirror is there to prevent some other excitatory light. Oh, okay, so it is just reflecting the green light and it is going to excite the object, right? But emission or barrier filter is there to make sure that there should not be any other lights, no other lights. Okay, if there is any, so this emission or barrier filter will block the other things, so it will only allow, in our case right now, is red. Okay, now this will come to the eyepiece and we will see this object like this. In this way we can see the object. Okay, I mean the emission filters allow only the fluorescent light that is emitted by the fluorescent sample. This is the sample to reach the eyepiece. So this is the actually basic components, or you can say, or basic structure. Okay, so what are the benefits then? So one benefit is high specificity. Okay, high sensitivity. Some specific wavelength of light is getting emitted, you can see here. Okay, so you can see some real-time imaging, real-time observation. Okay, so these things are benefits. There are so many benefits, maybe you can say, for fluorescent microscopy. For limitations, what you can say, this is benefits. Limitations, it may have photo bleaching effects. Okay, so the fluorophores can degrade over time with some continuous expose to the light, to the object. Okay, or it can be phototoxicity also. So prolonged exposure, normally these actually elute these things. Okay, so prolonged exposure can damage the live cells, right? So that can be called as toxicity. Okay, so we can say that these can be the limitations. Anyway, so the main thing is there will be one excitation filter, dichotic mirror, and emission at barrier filter. So it will allow, excitation filter will allow only one emission light, and it will be absorbed by the object, and the object will emit some different wavelength, and that can be seen by us. So this is the basic idea of fluorescent microscopy. Hope you understood. See you in the next video. Thank you.